The overall aim of this procedure is to estimate the relative affinity constant, Kb, of an agonist for the active state of a receptor by analysis of its concentration response curve. This is accomplished by first measuring the concentration response curves of a group of agonists for eliciting a response at a G-protein coupled receptor. The next step of the procedure is to estimate the Emax and EC50 values of the agonists using nonlinear regression analysis. These values are then used to calculate initial parameter estimates for the subsequent nonlinear regression analysis involving the operational model. As a final step, relative estimates of the affinity constants of agonists for the active state of the receptor are obtained using global nonlinear regression analysis of their concentration response curves. Ultimately, using a modified form of the operational model, the affinity constant of an agonist for the active state of a receptor, expressed relative to that of another agonist, can be estimated through global nonlinear regression analysis of their concentration response curves.
This method can help answer key questions in the drug discovery field, such as, does an agonist have selectivity for a specific signaling pathway? Generally, individuals new to this method will struggle because of the details of the mathematical analysis.
For estimation of relative agonist Kb values, a series of at least 2 agonist concentration response curves is required. A discussion of considerations